Hello everyone, thank you so much for taking time to listen to my presentation. My name is Varshini Suresh and I am currently an undergraduate student and I work on research at the Barnes Lab related to eDNA detection of various amphibian pathogens. Today I'll be discussing my current project entitled Collection Method Impacts Detection of Heterogeneously Distributed Amphibian Pathogens in a Playa Lake. So to start off, I want to talk about some background information that's really necessary to understanding my current study. To begin, we'll define what eDNA is. So eDNA is basically the process through which we acquire DNA from the environment rather than from a specific specimen. So by doing this, we're able to actually determine the concentration of the DNA of an organism, or in our case, a pathogen, in the environment and its presence in the environment. Additionally, it's important for us to mention the pathogens that we are working with, ranavirus and chytrid fungus. So as you can see from the top image, ranavirus can cause skin ulcers and internal bleeding in amphibians, which ultimately causes their demise. Chytrid fungus, as seen in the bottom image, causes a wide range of different symptoms. So you can have discoloration in the skin, which is pretty mild, or you can have hardening and peeling of the skin, which ultimately leads to the death of the amphibian. So there's a really wide range, but both of these pathogens, chytrid fungus and ranavirus, have actually been implicated in global declines of amphibian populations. And they've only recently been discovered in Lubbock, which creates a really big need for broader surveillance in this region. The novel detection of both these pathogens in the Lubbock area actually has led to our interest in this area and to this current study with a new question about what is the best detection method, and are these pathogens found in just one location or one type of location, or are they found just all around a lake or a body of water? So that leads us into our methods, which we'll talk about in the next slide. The first thing we did was we went out into the field to collect samples from Clout Park, which was a location in which ranavirus and chytrid fungus had been previously detected. We had to really consider testing locations because in order to determine heterogeneity or homogeneity of the pathogens, we had to test in locations around the lake. By doing so, we were able to get a good representation of different locations around the lake. This also allowed us to test different types of locations as well. In other words, we were able to see if human or domestic animal traffic was a correlating factor in determining where the pathogens were found. A map of the testing sites is seen in the image to the top left. At each of these testing sites, we actually used 3 different methods of eDNA collection. As seen in the table, we use precipitation, filtration, and sediment collection methods. And each of these collection methods is actually unique, especially in its sample size. For example, in the precipitation method, we had a set amount of water that we collected at each site, 15 milliliters. However, in the filtration method, the sample size was really variable across the sites, depending on how murky the water was, or depending on how much water we were actually able to get through the filter before the pores started to clog up. And in the third method, we actually collected sediment or soil instead of water. And 10 grams of the soil collected was actually used in DNA extraction, which was the next step. So we took all of our samples back to the lab and we extracted DNA from each sample. So for the first 2 methods, precipitation and filtration, we actually used a well-documented protocol detailed in the paper written by Faisatola et al. However, for the third collection method, sediment, we used a kit, the DNEZ PowerMax soil kit, and we used the protocol detailed within the kit. Once we were able to extract DNA, we then had to use PCR to see if the DNA of the pathogens was actually present in any of our samples. We'll start off with our results for the precipitation testing method. And before we begin, I'd like to say that sites L and sites N are actually not shown on the map. This is because while they were very close to site K, there was a GPS recording error in these sites, and so we did not show them on the map. Additionally, I would like to point out that positive detections for chytrid fungus are shown in blue, and positive detections for ranavirus are denoted in pink circles. We'll begin with chytrid fungus, which was detected at 3 different sites: Site A, Site F, and Site L. And as you can see, there were multiple positive detections at each site. However, for ranavirus, while it was detected in 2 different sites, Site B and Site D, there was only 1 positive detection out of the 6 replicates. Next, we'll look at the filtration method. Again, we can see that on the map, positive detections of chytrid fungus are denoted in blue circles, and locations of positive detections of ranavirus are denoted in pink circles. And just by looking at the map, we can already see that there's a difference in the number of detections of each pathogen. Chytrid fungus was more prevalent and was found at more sites, and was also found in more replicates at each site. However, ranavirus was only found in 1 replicate at 1 site, so we can already see that there is a huge disproportionality between the number of positive detections between each pathogen, but also between the 2 testing methods we've looked at so far. Lastly, we can look at the sediment extraction method, and for this method, it's important to note that there were only 3 replicates instead of 6 like the previous 2 methods. However, for chytrid fungus, there was at least 1 positive detection out of the 3 replicates. Since the project is still ongoing, we have not yet been able to run a PCR to determine if ranavirus was present through the use of the sediment collection method. This is something we will be working on in the future. Next, I would like to talk about some of the major take-home messages from the results. So we'll start with talking about chytrid fungus. And the best sampling method for this pathogen was the filtration method, because we were able to see a large number of locations with positive detections, but also a large number of replicates that had positive detections. Second, we could see that there was a heterogeneous distribution around the lake. And third, there was a very high prevalence of the pathogen, which was seen in the filtration method. Next, we can talk about the ranavirus results. And the best sampling method for this pathogen was actually inconclusive, because there were only 2 positive detection sites in the precipitation method and only 1 positive detection site in the filtration method. And the sediment method we have not yet been able to PCR, so the best sampling method was actually inconclusive. However, we can say that there was somewhat of a heterogeneous detection, because the sites in which ranavirus was found was pretty well distributed around the lake. And thirdly, the prevalence of this pathogen was low, and we can see this based on comparing our results for ranavirus with chytrid fungus. We were only able to really see 3 positive detections of ranavirus throughout the whole experiment. This leads us into some potential impacts. First, we'll start with some societal impacts, which are, for example, animal health and welfare and conservation efforts, which really go hand in hand. And the results of this study will impact and inform both conservation efforts and animal, or especially amphibian, welfare. Next, we'll talk about advancements in the field. eDNA has only been recently found in the West Texas area, and there is not really much known information about different collection methods. So the results of this study will really inform and impact advancements in this field. And thirdly, I would like to talk about commercialization. While there's not a direct impact from this study on commercialization, there's an indirect impact in environmental consulting and for making eDNA collection methods more effective and efficient. This then leads us to some future directions to consider. Firstly, completion of this investigation. There's a couple of things that we could do. First, we need to finish the sediment method testing for ranavirus and add more replicates for the kitchen fungus, just so that we are more confident in our results and the analysis that we can make from those results. Additionally, we can change the filter sizes for the filtration method because ranavirus is a particle that's smaller than the size of the pores that we used, which was 0.45. And by doing this, we'll make sure that we're actually catching more of the virus if it's present in the environment. Thirdly, we can do more replications of all of the different methods that we've used in this experiment. And so in this way, we can be more confident about the conclusions that we make. Some future investigations that we can consider are determination of pathogen survival in ephemeral soils. Because Cloud Park is a playa lake which dries up in the summer and then has water in the rainy seasons, it's important to consider how that change in environment affects the presence of the pathogen in the environment. Additionally, this study can inform amphibian population conservation efforts in the future as well. Lastly, I'd like to thank a couple of people without whom this project would not have come to fruition. Firstly, I'd like to thank my PI, Dr. Matthew Barnes, for all of his help and guidance with this project. I would like to thank Beth and Mark for helping me collect my samples. And then I would like to thank the Pi² program and the Undergraduate Research Scholar program for granting me an opportunity to pursue my interest in research and for giving me a platform in which to do so. Additionally, I'd like to thank the Honors College for all the support and advice that the advisors have given me in pursuing my research goals. Thank you so much for listening to my presentation today. If you have any questions or want more information, please don't hesitate to contact me by email. Thank you so much for your time.